Good morning and welcome. Thank you for being here and for making the time. This is a presentation of the Dave and Doris Leaning on Sustainability series, and it's co-hosted by Great Pond Foundation and Chilmark Pond Foundation. We're really excited today because we have Dr. Javier Lloret of the Marine Biological Laboratory in Woods Hole, and Javier has a whole bunch of data to share about some of our beloved ponds and what's causing the impairment. We all know nitrogen is the main culprit, but where is it coming from and what's the source? So he's going to let us know what he's been finding with his team over the last several years. To all the folks who are on the Zoom who've given us access to their properties to collect, thank you. We're grateful. Please keep your microphones off for the remainder of Javier's presentation. At the end, we'll take questions from the room, and then we'll take questions from the chat and Zoom. And at that time, you'd be welcome to put your microphone on. Javier, we'd be grateful if you could get us started.
Yeah, thank you very much. Let me share my screen so I can share a few slides with you guys. Yes, that is taking a minute or two. There we go. So, well, thank you all very much for being here today and to listen to this presentation that I put together. I mean, the whole intention of the presentation today was to give you guys a little overview of some of the things and some of the results that the work that we have been doing in several of the vineyard ponds for the last few years. And what I tried to capture with the title of the presentation, focusing on addressing some of our coastal pond challenges, and more specifically focusing on the problems that are derived from excessive nutrient inputs and excessive nitrogen inputs more specifically. So the way I organized the presentation today is that at first I'm gonna just give you a little bit of an introduction on some of the concepts that we're gonna be covering later on in the presentation, and then I'm gonna jump directly into my results from the analysis that we did in the last couple of years. So you get a better sense of what the data looks like and what we have learned in terms of these challenges that we're seeing in our coastal ponds. So without further delay, I always like starting this presentation by pointing out something that, maybe it's not that obvious for some people, but most of our communities, including the communities here in the vineyard, are facing what I like to call this nitrogen dilemma. Nitrogen is an element that is essential for life. All life forms need these elements to build their bodies, and we need nitrogen to grow our food, as is shown in the image here on the right.
Can you speak up just a little?
Yeah, absolutely. So it does have very clear benefits to us to have access to this element, to nitrogen. But it also has some drawbacks. And one of the most dramatic drawbacks that nitrogen can have is that when we put it in excess, it can cause a degradation of the environment. These problems, when it comes to the coast, and when it comes to our local estuaries and coastal areas, like the coastal ponds here in the vineyard, can lead, the excess nitrogen that we put in the environment can lead to a cascade of events that we scientists like to call eutrophication. It's a terrible word. I know that is jargon, but in reality, I think that most of us in this audience, including the folks here in the room, as well as most people on Zoom, probably know exactly what I'm talking about. I'm talking about algal blooms like the ones here depicted in the photos. I'm talking about the water turning green. I'm talking about an overall decrease of the water quality of our coastal ecosystems, of our coastal ponds, that can lead to other effects, like, for example, losses of valuable habitats like seagrass loss, eelgrass loss. And it can also lead, in some instances, when the process gets really out of control, it can lead to decreases in the oxygen level in the water and the appearance of these dead zones, or with fish kills and shellfish kills. And this is definitely not where we want to be. We want to stop this process before it even starts, if possible. And as I said, this is not a problem that is only unique here in the vineyard, but it's a worldwide problem. It's a very widespread problem associated to the development of the land, intensification of land uses, that we humans put a lot of nitrogen into the environment, resulting in this environmental degradation that I am calling here eutrophication. So this is a very widespread problem that we humans in general are dealing with in many parts of the world, as is depicted in the map on the right, with all those dots indicating locations around the world that have suffered the consequences of this environmental degradation. The next slide was just to kind of remind you guys, or kind of explain to you guys in very simple terms what happens to our coastal ponds when we add excess nitrogen. So in the first drawing here on top, it's just like a little profile of one of our coastal ponds. And in those pristine situations in which there are no humans around, and kind of thinking of how the ecosystem looked like before humans colonized the land, those places tend to have a very high environmental quality. The waters tend to be very clear and transparent, and that allows for the colonization of the bottoms of those shallow ponds by valuable habitats like eelgrass beds. The little guy here on the right is very happy for a reason. This is a habitat of very high environmental quality. Now, what happens when we add nutrients in excess to this type of ecosystem? Well, one of the first things that we see is that those eelgrass beds start disappearing, and they get substituted by competition by opportunistic macroalgae. The algae is growing, feeding on those nutrients that were put in there in excess, and displays the eelgrass from the bottoms. And in the water column, I don't know if you can see these little bubbles that I put here. Those are like phytoplankton cells. The small microscopic algae also grows, feeding on that excess nitrogen and nutrients that we're putting in the water. This is a habitat of much less environmental quality. So you can see that our little friend on the right is not that happy anymore. If we keep adding nutrients even further, what happens is that all the algal growth that we were seeing before, it becomes like completely out of control. Excessive macroalgal, big algal masses in the system, excessive growth of phytoplankton, of the microscopic algae in the water. The water tends to turn green. The waters tend to decrease in their transparency. And in some instances, especially by the end of the season and with high temperatures, all that organic material that is being produced in the system starts to decay. The bacteria that is chewing in all that material consumes the oxygen, and that can lead to fish kills and shellfish kills that in some instances we can even see in some of our coastal ecosystems. So we really want to stop this from happening. We don't want to get to this situation kind of at the end of the line of this process that I call eutrophication. We want to stop it before it even happens. And to do that, we need to make sure that we manage that excess nutrient that we're putting into the ecosystem. Where is all those nutrients coming from? Well, unfortunately, it's us. It's our activities on land that cause excess nutrients to be delivered into our coastal ecosystems. And it comes basically from three major sources that I'm highlighting here in this next slide. On one hand, the inputs of wastewater coming from our houses contain a lot of these nutrients, nitrogen, and eventually those nutrients make their way into our coastal ecosystems. The second very important source is the application of fertilizers, whether it is for agricultural purposes or for the fertilization of our lawns. We put a lot of fertilizers into the environment, and those nutrients that are contained in the fertilizers can also make their way into our coastal ecosystems. And the third source, and probably one that is sometimes a little overlooked, is the pollution of the atmosphere. The combustion of fossil fuels put a lot of nitrogen compounds onto the atmosphere that eventually also get deposited onto the surface of our ecosystems, mostly through precipitation. And what happens with all that stuff that we put on the surface of the land, whether there is wastewater deposition from the atmosphere or fertilizer, all those nutrients get on the surface of the land. And here in this particular landscape that we are here in the vineyard, and also most of Cape Cod, those nutrients get transported by the water that comes with the precipitation, and infiltrate into our soils and flow into our coastal estuaries, mostly via groundwater. There are a few streams that carry some surface water and some runoff, but for the most part, most of the nutrients that we receive in our coastal ponds come via groundwater. So that's going to play out a very important role when we're trying to figure out where the nutrients are coming from, because we're going to need to look for them underground. So with all these things in mind, we put together a project, my lab at the Marine Biological Laboratory, in collaboration with the Great Pond Foundation and the Chilmark Pond Foundation, a project that we entitled Laying the Foundation for Coastal Pond Restoration, and a project that, in my view, has four major objectives or goals that we're trying to address, keeping in mind what our ecosystems, what our coastal ecosystems need as they are facing this increasingly problematic issue of nitrogen pollution. The first goal that I am highlighting here is the efforts that the Marine Biological Laboratory is putting together to assist both foundations in their ongoing efforts with water quality monitoring. And so we're putting the disposal of the foundation, our facilities, and our personnel to do the analysis of nutrients and pigments in our facilities and with our equipment there at the Marine Biological Laboratory. But we also have very major goals that I summarize here, and these three other goals here that are, in the first place, identifying major nitrogen inputs entering our coastal ponds, where and how much nitrogen is entering our coastal systems, and also identifying the sources, what activity is putting there that nitrogen. So that's going to lead us to understand a little better what the problem is and how we can start to solve it. We're also putting a lot of attention on the effects on ecosystems and the effects that this excessive nutrient inputs can have on our coastal pond from a more biological point of view. And the third objective, that in reality is more like an overarching objective, we have a very clear focus on solutions. So all the scientific activities that we're putting together within the framework of the project have that overarching goal of trying to understand what's going on and how we can propose solutions to remediate the problem to the major extent possible. So it's providing science that could inform management and policymaking. So with that in mind, and having nitrogen as a central piece of what we do, we have been conducting over the last couple of years, for the most part, a lot of sampling in some of our coastal ponds here in the vineyard, doing a lot of groundwater sampling, trying to identify these nutrient sources, doing a lot of work in the ponds themselves, collecting hundreds and hundreds of samples and looking at the problems related to nitrogen pollution. And we carried out a lot of analysis of the nutrient contents in those samples, and even using some innovative tools to identify sources like the stable isotopes analysis that I'm highlighting here in this slide as well, that I'm going to tell you a little bit more about later on, but it's one of the tools that we use to identify where the inputs of nitrogen are coming from. So the first group of slides here, I'm going to show you some of the results that came out of these analysis that we did at quantifying the nitrogen inputs to our coastal ponds. So it's mostly, for the most part, the analysis of groundwater samples that we collected in several of our coastal ponds. To do this, we visited two of the ponds here in the vineyard, the Edgartown Great Pond and the Chilmark Pond. And the sampling design that we carried out was that we visited the periphery of these two ponds and along the shoreline and established like these transects parallel to the shoreline of some of the major coves that are flowing into the ponds themselves, and collected samples approximately spaced like 10 meters in between samples. So one of these transects can be like as long as 100 meters or so. And we collected samples of groundwater that is immediately flowing underground right at the point of entry of those waters into the pond. So we are going to be able to capture the nutrients that are right at the moment when they're entering the pond. So we are going to be able to quantify how much nitrogen is entering the pond in different locations along the shoreline of the different ponds. We did this, and this is an ongoing work. I mean, we have collected a ton of samples, more than 250 samples so far, but we are continuing to cover ground because it's quite large ponds, as you can imagine. This takes a substantial amount of effort and time to do. But we collected these samples with these devices that I'm showing here in the picture in the center. They are portable well point piezometers. This basically, to explain it in simple terms, it's just like a rod, a hollow rod that you poke into the ground, and you are capable of reaching that groundwater that is very close to the surface once it's so close to the pond. And by pumping out, creating vacuum, we can pump out the groundwater before it reaches the pond, right? We collected our samples and analyzed the nutrient contents and the isotopic signature that I'm going to tell you later on a little bit more about of those inputs. So that's basically what we did in these two ponds. We visited a lot of locations, but we centered our study in some of the major coves that are flowing towards the two ponds. And these little rectangles kind of indicate the areas that we have visited so far, covering most of the periphery of both ponds, Great Pond and Chilmark Pond. So what did we find? Well, let me show you some of these results, and I'll try to explain. Of course, I'm not going to show you results from 250 samples. I don't want people falling asleep here or on Zoom, but I'm going to summarize some of the most striking results that we got from the analysis of all these samples. We did find a lot of evidence of the presence of septic plumes. Now, what do I mean by that? So, this graph, what it's showing is along one of my transects of approximately 100 meters, the concentrations of nitrate present in the samples that we collected. So each one of these dots is one sample, right? Now, what you can see in the graph is that the concentrations, for the most part, were relatively low. That's the background level of nitrate for this particular cove in this location. But then all of a sudden, in some samples, we find these super high concentrations, like several orders of magnitude higher than that baseline. This is a pattern that I have observed everywhere around, also around Cape Cod, where I do this analysis, and when there are a lot of septic systems in the watershed. Every time there is a house nearby, kind of upgrading upland, the septic system, the individual septic system of that house releases a lot of nitrogen into the aquifer. Now, you may think that once that plume of nitrogen reaches the aquifer, everything gets mixed. But in reality, it doesn't work that way. Water in the aquifers do not mix horizontally very well. And so that plume of nitrogen travels almost at a constant concentration all the way down into the pond. So when I sample right at the shoreline, we can be seeing like very, very low concentrations. And all of a sudden, when I hit a septic plume, we did find a concentration that is several orders of magnitude, what you would expect in that area. So this is a clear indication of the presence of septic plumes. I'm not showing it here, but if I showed you, actually, let me go back. it's actually from this pond over here. I hope that everyone can see this, but you see these two houses, one in the beginning of the transect and one towards the end. If you look at this graph, that's probably that house, the septic system corresponding to that house corresponds to this peak, and the septic system of the following house along the road is probably that other peak. Well, this is kind of at the small scale, at the scale of a transect, but the picture becomes even more interesting when you look at the pond as a whole, because we were capable of identifying that in reality, despite that the whole pond is what's suffering the consequences of these nutrient inputs, most of the inputs are actually taking place through very specific parts of the ponds, right? In the case of Great Pond in Edgartown, the Meshacket Cove and the Slough Cove are responsible for more than 70% of the nitrogen inputs. The numbers that I'm showing in these boxes are the concentrations, the current average concentrations of nitrogen in the samples that we collected in those coves. When you multiply that by the amount of water that is flowing there, you get a total amount of nitrogen. And when you look at the whole pond in terms of how much nitrogen comes from which cove, you can realize that the Meshacket Cove and Slough Cove represent 75% of those inputs, that the upper ponds in Chilmark Pond represent more than 50% of the inputs. To get to that 70%, we can add the Waits Cove in Chilmark Pond, and we will get most of the inputs that are entering Chilmark Pond. So this is a very important information in terms of management, because if we want to solve the problem for the pond as a whole, maybe a good place to start would be these particular locations, because most of the inputs can be controlled if we address the problem starting at least in these particular locations. The next group of slides, I'm going to be, so far I've been talking about total amounts of nitrogen, amount of nitrogen that is entering our ponds. But remember that one of our goals was actually knowing where is that nitrogen coming from? Who put it there in the first place? And to do that, this analysis with stable isotopes, and this is some of the equipment that we used there in my lab, in the Marine Biological Laboratory. And it's a very interesting tool. It's a very interesting tool because we can take advantage of a property that the different sources of nitrogen have with respect to their isotopic signature. It is the fact that the different sources have very, very distinct isotopic signatures. I'm showing in here the values of those signatures here with this notation, delta 15N. That's the isotopic signature of wastewater. It's a very high number. You don't need to worry too much about numbers. I'm definitely not going to bore you with chemistry. But what I want you to understand is that we can use these signatures of the three different sources, wastewater, fertilizer, and atmospheric deposition. We can use them almost as a fingerprint that these sources leave behind when they put nitrogen into the environment. So by looking at the isotopic signatures of the inputs that I'm measuring, I can figure out whether it came from wastewater, whether it came from fertilizer, or if it's just pollution from the atmosphere. So that's how we use this isotopic information. So, well, what do the results show? Well, it was maybe not surprising because we knew a lot about these places, but it was really nice to see that it really worked at identifying the nitrogen sources. This graph is showing the isotopic signatures of the inputs occurring through the Meshacket Cove in Great Pond and the Slough Cove, also in Great Pond. And what you can see for the values of the Meshacket Cove is that are a much higher value than the inputs that occur through the Slough Cove. Now, these lines over here, these blue lines, these dashed lines, indicate the expected isotopic signature of the inputs if they were just wastewater, like almost like pure wastewater, and what you would expect to see if those inputs were fertilizers or atmospheric deposition. the inputs in the Mashakit code are clearly dominated by inputs of wastewater, which is not surprising because that code is receiving the inputs of the Edgartown wastewater treatment facility. So there's a lot of wastewater coming through that location. The signature in the SLAW code inputs, it's kind of in between the two sources. So what this tells me is that there's a lot of wastewater entering through that code, but there's also some contributions coming from fertilizer and potentially some from atmospheric deposition. So it's more like a mix of source in this code. This is not telling us anything about concentration. I already measured that. This is telling us about the origin of that nitrogen. Where is it coming from? So in the case of Mashakit, mostly wastewater. It shadows all the other sources because of the amount of wastewater. And in SLAW code is a mix of the two. It almost looks like 50-50, fertilizer and wastewater. We did a little bit more with this isotopic work because we wanted to figure out, well, what happens with all the nitrogen once it reaches the pond? Once the nitrogen reaches the pond, the plants and the algae are going to start using that nitrogen, and they're going to start growing, and sometimes they can grow out of control. Sometimes we even see like undesirable cyanobacteria blooms that are feeding on that nitrogen and can be toxic, right? So we wanted to figure out, well, or get an answer to the question, what nitrogen are those algae using to grow, right? So we did an analysis of the isotopic composition of the phytoplankton of the microscopic algal species that are growing in the pond, in trying to figure out what kind of nitrogen they're using to grow. And it turned out that in both of these ponds, more than 50% of the nitrogen that the plankton is using to grow, and sometimes grow out of control, actually comes from wastewater. Immediately followed by fertilizers as the second most important source of nitrogen. So, well, what next? Well, one thing that we really want to get a hold on is to try to really get a good number and understanding how much nitrogen is reaching these ponds. We're doing a very big effort put into sampling and measuring things, but we also need to understand A, where is it coming from, and B, how do we solve the problem? So it would be great for us to have a better understanding of how the nitrogen is coming from the land into the ecosystem. Right now, the only pieces of information that we have available to do that are the data that we are collecting at this moment and the data that was produced during the MEP effort for both ponds. Now, I think I got the dates right, but we're relying on information that comes from 2008 for the Great Pond in Edgartown. That was the date of publication of the MEP report for that pond, and 2015 for Chilmark Pond. In both cases, we are relying on information that is at least 20 years old, because these reports were put together with data on land uses, data on the different sources of nitrogen that dated before the reports were published. A lot of things changed in 20 years. So what we're proposing to do in this next year, in collaboration with both foundations, is to carry out a new effort on modeling the natural loads to both of these ponds and update the available information in terms of how much nitrogen are we putting there and who is putting it there, right? And so to do that, we're going to use a peer-reviewed model called NLM, a model that was developed in this region, this specific region, to answer this kind of question. It's a peer-reviewed model. It's publicly available. It has been validated against real data from this very location. It allows us to partition the nitrogen inputs among the different sources with water, fertilizer, atmospheric deposition. And it even includes some measurements of uncertainty. So we're going to be able not only to tell how much nitrogen is coming from the different sources, but how sure we are that that number is correct. if that makes any sense. We're gonna use the most updated land use data rather than rely on data that dates more than 20-something years old. And we're gonna update to the values for the current nitrogen sources, including updated atmospheric deposition. I'm gonna make a little parenthesis here, but atmospheric deposition in the last 25 years has decreased by half. That's something that was definitely not included in previous model assessments, right? And so we're looking at a picture that has not much to do with what is happening today. So that's one of the things that I'm planning to update. We're gonna update the data on fertilizer usage in these two watersheds. And we're also gonna update information in the number of septic systems that have probably changed over the course of 25 years. New houses may have been built. Some other houses may have been connected to a centralized wastewater treatment facility. Some septic systems may have been upgraded. So there's a lot of information, wishful thinking, right? But there's a lot of information that I guess that in 25 years has changed quite substantially. And so we're planning to get an updated number for that. And we also have like an excellent opportunity here because we're doing a lot of sampling. So we're gonna be able to validate this modeling effort with our own measurements that we're collecting in parallel to this effort. So that's gonna be a very interesting part of the work. And something that is keeping me very excited, which is that last point that I have in this slide. This effort is gonna allow us to do some scenario modeling. What do I mean by that? What I mean by that is that once I have the model set up, we're gonna be able to test and see and discuss about different management options. What happens if we do X or Y or Z? How much is the natural loading gonna change to these ponds if we do this or that or the other thing? So I think that that's a very exciting piece of work because it can be used as a tool that can inform management and also policy making. Well, we've talked a lot about nitrogen and how bad it is and how much nitrogen is reaching our ponds. But I always like to end my presentation with a little bit of some positivity. And so, as I said from the very beginning, some of the efforts that we are carrying out within the framework of this project are also focusing on solutions. And one of the things that we're exploring a variety of options, right? But one of the things that we have been working on in the last year, and I prepared a few slides about it just to show you guys some interesting results, was this examining this potential role that coastal wetlands, freshwater wetlands actually play at intercepting some of the nitrogen that is coming from land. Naturally, you guys that live here in the vineyard are probably familiar with this kind of landscape that I'm showing here in this photo. This is one of the heads of one of the coves. I do not remember which cove this one is, to be honest with you, but I'm pretty sure that you're familiar with this kind of landscape. It's the head of one of the coves that flow into Edgartown Red Pond. And what you can see here in the background is that tree line, that forested area that we have in many areas here in the vineyard.
This is Mashaka Cove.
It's probably Mashaka Cove. It does look like. Anyways, before the groundwater reaches the pond, in some cases, in many of these coves, we have this area of very densely vegetated areas of freshwater wetlands, of plants that are growing very densely and very green around these upper areas of the coves. And in many cases, they're also intersected, by these little tiny creeks that are groundwater fed, right? The groundwater is flowing and feeding these little creeks that is actually the water course that delivers all that fresh water into our ponds, right? And the nutrients it contains. Basically, now looking from the side, what I'm saying is that this can be the area of land on the left, and this is that little creek that I was showing in the previous picture that flows into the ponds themselves. We have this ecosystem in between, these freshwater wetlands that in a way are intercepting the flow of fresh water that is happening underground. So theoretically, these plants may be growing using the nutrients that are flowing underneath, right, underground, and potentially taking up some of that excess nutrients. So we wanted to examine the role that these plants are playing, whether or not they are taking up some of that nutrient. So those are the results that I'm going to show you next. Basically, the sampling design that we did, and I think that this is a photo of the Mashaqet code, the head of the Mashaqet code, that is probably corresponding with the photo that I was showing you before. But basically, we collected samples along this yellow line that I'm calling here the tree line, right? We collected sample within this wetland, which is that very dark green area that I have in between the yellow lines. And we also collected samples in this little creek in blue, that is the one delivering the fresh water into the main Mashaqet cove and into the Edgartown Great Pond, right? So we are interested in seeing how much nitrogen gets intercepted in the groundwater as it travels from the tree line into the wetland and into the creek, if that makes sense. So that's what these results tells us about. So I'm going to show you in all these graphs the data that we see on and how it looks like on the tree line, in the wetland, and then in the creek. The graph here shows the concentration of the different nitrogen compounds that we found in the groundwater. And what you can see by the length of the bar, it decreases very dramatically as it travels from the tree line into the wetland and into the creek. If you quantify the decrease, it corresponds to a 42% decrease in the nitrogen that is flowing through the wetland. There's some nitrogen that is not flowing through the wetland. I want to remind everyone that before we get too excited. But at least when you have these wetlands present, the amount of nitrogen that goes through them can be reduced up to 42% in this scenario. Not only that, but if you look at the colors on the bar that indicate the different forms of nitrogen, the nitrogen is also being transformed. The inorganic forms, nitrate and ammonium in blue and yellow, those are the species of nitrogen that are more prompt to cause algal blooms, because the algae loves this kind of nitrogen, right? Now that gets raised like very, very nicely. And part of that nitrogen gets transformed into this green area over here, which is DON, dissolved organic nitrogen. Now, those organic forms tend to be a little bit more difficult to chew on by bacteria as well as by the plankton. So not only we are getting rid of most of the nitrogen, but we're also changing its nature so it's not as easily accessible for the plankton to grow. So this is a very exciting role that these freshwater wetlands are playing, and they're happening there naturally. We got some other interesting results by looking at the isotopic signature of the nutrients as they move from the tree line into the wetland and into the creek. And what you can see in this graph is that the isotopic signature was very high in the tree line, decreases in the wetland, and becomes even smaller in the creek. So the wastewater signature, if you remember from previous slides, the wastewater has a very high signature and the other sources have not. The wastewater signature of those inputs, it's being erased. It's being removed partially by these wetlands. So indicating that the wetland not only is removing nitrogen, but it's taking up some of that groundwater nitrogen that I was telling you about that was very contaminated by wastewater. And we went even further. We collected a few plant samples and analyzed the isotopic signature in the tissues of the plants that are growing in the wetland. And we came up with this very exciting discovery that not all plants in the wetland are playing that role. The two first species here, the reed and the skullcap, reflect the signature of the wastewater inputs. So those two plants are capable of tapping into the groundwater and taking up those excess nutrients. We have two other plants here, the sweet gale and the wort, that are not taking up that nitrogen. So these two plants are probably just growing on the nitrogen that falls with the rain on them, right? Instead of tapping onto the groundwater. And we have another three plants here that are probably doing a little bit of both. They're feeding on some of the groundwater nutrients, but they're also feeding on the rain nutrients. So it's very exciting to see this data because it's telling us a lot of information about A, how the wetland behaves in terms of removing excess nitrogen, and B, which plants are responsible for it, which can be very interesting in terms of management because we can potentially make an effort on restoring some of these wetlands, but we need to be careful with which plants we put there. We want them to be able to play this interesting role at intercepting nutrients. So yeah, with that, I'm gonna finish just by kind of summarizing some of these findings, just as a final summary of reminder. I'm basically just gonna read this, but I just wanted to highlight some of our major findings with this work. Our groundwater nutrient sampling clearly revealed the presence of these septic plumes, and they occur all over the periphery of both of our coastal ponds. So it's an important source that needs to be taken into consideration, the septic source. Very interestingly, the majority of the nitrogen inputs that reach our ponds actually come from very specific locations along the shoreline. It's not all over the place where we need to look for the major inputs. There are some coves and some particular locations in these two ponds that are responsible for the majority of inputs, which is going to have implications for the design of effective management strategies. We did identify that wastewater is probably the most important source of nitrogen, but it's immediately followed by fertilizers. So there's also a little bit of a problem with fertilizers in these ponds. And last but not least, as we saw in the last slides, the wetlands, the freshwater wetlands show very high potential for attenuating nitrogen loads, which can be used or managed if we want to apply restoration initiatives to improve the natural capacity of the landscape to absorb some of these nitrogen inputs. So yeah, with that, I think that I'm going to leave it there, and I would like to take any questions that you may.
Yeah. I can bring up the bases. Thank you. And perhaps we could start with the room. Does anyone have any questions in the room? Okay.
So you were talking about the value of freshwater wetlands.
Mm-hmm.
Is there a similar value to wetlands adjacent to the marshes like saltwater?
Yeah, absolutely. So that was, I mean, most of the work that I have been doing so far has been with tidal wetlands and salt marshes. And I was pretty aware of their role at intercepting nitrogen inputs coming from land as well. So they are very good at doing that.
Are there the same way you broke down the different species, are there specific species that do a better job that you could share with us?
I do not have those data with me, but I can dig it out. I do have a lot of information. I can certainly dig it out. But one of the things that came out from this study, as well as for some of the studies that I've been doing on salt marshes, is that the root depth seems to matter quite a bit in here. So many of our salt marsh plants have different root length, but most of them are relatively shallow. I'm especially interested in those species that are closer to the upland, right? And if they can tap into the groundwater, because some of our salt marsh plants that live at lower elevation are probably feeding on the nutrients that come from the pond water or from the ocean. So I'm interested in the ones that actually live closer to the land. And in that instance, root depth, I think that really matters. And that's something that we saw in this data as well. Because for example, the Phragmites, the dreaded Phragmites that no one likes, in reality has a very deep and very widespread root system that is capable of tapping into that groundwater. So there's, again, one of those trade-offs between, what do you do to manage your wetlands and what outcome you want, and, in terms of, promoting the restoration and remediation of natural roads. If you want to have a certain type of wetland with native plants and stuff, you really would like to manage the phragmites, right? But if you look at the data coming from the nitrogen retention capacity of the different plants, having the phragmites, at least for a while, may help you, retain at least some of those nitrogen inputs, while you put in place other measures to address the nitrogen issue.
And are there native plants that have that same capacity and deep roots?
Yeah, I do think so. I'm not a botanist, I have to confess, unfortunately, but I am very interested now in learning a little bit more about which plants specifically are the ones that are carrying out, these or playing this role at intercepting nutrients, because that's a very clear indication in the isotopic signatures that some plants do it, some plants don't.
Are there other questions from the room? Because you do have people in the chat that are excited to share stuff too.
Can I ask a quick question, which is maybe a silly one? I feel like often people are asking, well, how much does it matter where like, when they're thinking about their individual inputs, where I am in the watershed, whether I'm close to a coastal pond or whether I'm way upland? I'm imagining based on what we just saw, it has to do with everything that's between them and the water, like how much vegetation, what type of vegetation, how many more houses, how much is adding to their, but is there any sort of general answer to that question, or?
So it's definitely not a silly question, but a really good one. The way I like to see it, when I look at the total inputs, I look at the big number. So everyone plays a role on the nutrient pollution issue. So we all contribute kind of the same, right? Now, when I presented the data on the specific codes that are receiving most of these inputs, what I'm thinking there is on the possibility of applying some remediation or addressing the problem in this particular codes just to be able to stop the problem. But the problem is widespread all over the periphery of the ponds, because at the end, the nitrogen is going to reach the pond and it all gets mixed and everything contributes to the problem. So the problem needs to be addressed at the entire watershed scale, but some remediation strategies can be applied in a specific area that kind of channel all those inputs through. So yeah, I think unfortunately we are all to be blamed for the problem, not just some specific people that live in specific parts are causing more trouble than others. The reality is that we all put approximately the same amount of nitrogen, and once it reaches the pond, everything gets added up.
With regards to the isotopes, where does fresh animal waste fall into the isotopes?
Freshwater and
Sorry, fresh animal
Manure.
waste, like manure
Oh.
and urine coming from like livestock on the land.
how do you mean from natural populations, from birds?
I
and
was
other-
thinking more like farm animals.
Oh, you're thinking of farm
thinking-
animals? Yeah, so we didn't test specifically for farm animals. I mean, there are certain number of them. In terms of their isotopic signature, it can be something closer to the wastewater that we see from humans, but a little lower.
Would it matter if it were, so like farm animals that we're thinking about, like sheep, cows, horses, they're mammals, whereas if it was more about, yes, wild birds.
Yeah,
then
like
would
chicken.
those have two different signatures?
It does matter
slightly?
because it does matter. So it matters because most of the manure that many farm animals put out there, like cows, I'm thinking of cow, sheep and stuff like that, is mostly in an organic form. Now that nitrogen needs to be chewed by bacteria. It needs to go through the soil and some other bacteria are going to do their thing there. It needs to travel all the way down into the aquifer, and then it gets almost transformed, immediately transferred into our coastal ecosystems. Now, in those organic forms, they're difficult to be used by the plants, right? Now, the manure coming from chicken has the potential to be transformed into inorganic forms more rapidly. It produces a lot of ammonia. Now, algae loves ammonia, right? And so it does matter which animal is putting the manure there. But what it really matters, if you want a more direct answer, what really matters is the amount, right? Like the total amount of nitrogen that is contained in those, that manure is what really matters. If we're putting a lot, if we have really big farms, it's gonna be a big problem. If we have a few animals here and there, it might not be that much.
And I think we're getting a lot from the chat, so it might be good for us to check in there. So let me, let's say, Earl Barnhart said increasing nutrients inputs into wetlands will increase plant growth and eventually the wetlands will become drier lands and other species will be able to be established. So maybe that's more of a comment. I didn't know if you had any thoughts on that.
You mean the wetland transitioning as we are promoting plant growth? Yeah, I mean, this is not a long-term solution, obviously. Like the problem of the nitrogen pollution in these ponds needs to be addressed and cut from the origin. We cannot rely on the natural capacity of the ecosystem to process this nitrogen only. But it does show potential to, as we are dealing with our problem, which is a big problem, will require time to be fixed. We can take advantage of some of the things that nature does to at least lower the temperature a little bit. In the long term, as the person on Zoom commented, if we keep feeding nitrogen to a wetland, eventually, the wetland may transition into an upland area that maybe is not going to be as effective as intercepting nitrogen as it is right now, when it's right at the interface between the soil and the fresh water that is flowing right underneath.
So
So,
some
yeah,
of the
we-
solutions are short-term and some of them are longer
Yeah.
term that we have to stop.
That's right. When I'm talking about short term, in terms of the transitioning wetlands and the effect that our viewer is talking about, I'm talking about decades, right? So it will take some time for the wetland to gain enough elevation so the plants are not capable of tapping into that groundwater anymore or any longer. But I mean, it's a mid-term solution. Let me put it that way, not short-term, but mid-term solution.
We also have a fantastic presentation comment from Hui colleague Mindy Richland. Peter Grunthal says, have you established any possible unintended connection between Mashakit Cove and the Edgartown wastewater treatment plant?
Unexpected?
Unintended is the word.
Well, that cove, I mean, unintended, that cove receives the inputs. Fortunately, the plant is doing an excellent role at removing as much nitrogen as they can, but eventually the nitrogen is going to come there. As we connect more houses to the centralized facility, we're going to continue to receive more and more nitrogen into the pond. So it's not unintended necessarily, it's just like, it's what it is,
It
right?
is what it is.
It is what it is.
From Liz, if septic system is contributing to the nitrogen loading, what can be done to reduce this? So I guess individual septic systems.
Yeah, well, the towns are doing a lot of work on this, as we probably all know. I mean, the traditional approach is just connect people to a centralized water treatment facility and get that nitrogen out of the system. But we all know that that's extremely costly. And for many people and many families, including mine, I do have a septic system in my house that is currently out of the question.
There are many IA systems to innovative
Yeah.
alternatives.
That's what I was gonna comment on next. So there are some alternative systems, septic systems that are extremely effective actually at removing nitrogen. And I'm talking like more than 95% of the nitrogen of our waste is being removed in this innovative septic systems. They are currently pricey. And so there's a little bit of pushback on that, understandably. But I do have a lot of hope in the technology, because I think that as we invest and we're showing more interest in these particular technologies, the prices are definitely going to go down. And one would hope that it will be more affordable in the future, and that the towns and the neighbors are going to be able to actually, put one of these systems that can be as effective, if not more effective, than a centralized wastewater treatment facility.
Okay, we're gonna take one more question from the chat and then encourage people in Zoom, if they want to raise their hands, we'll call on them. So I think we have Sakiko's question, then we'll call on Steve Leuenberg and go from there. So Sakiko asks, can we say a robust growth of Phragmites is a sign of septic sourced nitrogen leakage?
Yes and no. So when you find, Phragmites, this species, as some of you probably know, it's an invasive species, and this particular variety is just less sensitive to salinity than others. So it proliferates like in many of our ecosystems here in the vineyard, as well as on Cape Cod. It usually indicates that there is a freshwater source. Now, this species likes nitrogen, right? It's opportunistic in growth. It takes advantage of the presence of fresh water. But if you have like a root source of nitrogen, the plants are going to grow like pretty healthy. So, I mean, their presence doesn't necessarily indicate that you have a big problem with nitrogen, but their health may be an indicator that, you probably have like a few plumes of high nitrogen coming from them. If it's becoming like increasingly green and tall and healthy looking, it's probably an
So
indicator.
beautiful Phragmites could be an indication. Okay. So Steve, did you have a question? If you unmute, you can ask it.
Yes, the question is, to what extent does salinity affect this whole issue of growth of nitrogen-sensitive material? Specifically, the effects that we have of opening the ponds periodically. How does that affect this whole issue? And can that be part of the solution?
Yeah, thank you, Steve. That's a really good question. It's one of the things that we're looking into, actually, in collaboration with both of the foundations. So yeah, the regular openings of some of our ponds, the ones that are isolated from the ocean, can be actually like a pretty effective way of getting rid of the nitrogen, because you're literally flushing them out, right? What you really don't want is those nitrogen compounds to build up in your system. So if we regularly open the ponds to the rest of the ocean, we're flushing out a huge amount of nitrogen out of the system, and therefore preventing some of these, undesirable algal blooms that we are seeing in some of our ponds. Now, some of the results that we have been working on, and the foundation are doing a lot of work on that too. Some of the results point out to the fact that both the timing as well as the duration of those cuts, of those inlet openings, really matter, right? Because you really want to be able to flush those nutrients out, and it does take a while for some of these ponds, because the tides bring cleaner water in and take dirtier water out, but it does take a while to flush the entire pond. So you really want to make sure that those pond cuts are effective and that they stay open for a certain amount of time, and at a specific times of the year when you really want to flush the nutrients out right before the algae is taking it up. Right?
Javier, would it be safe to say that not every pond cut is the same, and that we need to pay attention to not just time of year, but how much volume of water, sort of how big the tidal chain exchange is for the cut and the duration of that.
That's exactly right, right? So the hydrodynamics here like play a very, very, very major role. Controlling, how much water we actually flush out and how much nutrients that are dissolved in that water we can actually flush out. So yeah, it does matter a lot.
Are there any other questions from the audience? You can feel free to raise your hand or unmute and you can take, oh, there's apparently some in the room. I guess you guys have to wait.
Can I ask a follow-on to that one, which is, I've heard recently that when the pond has been opened and there's draining, it accelerates the input from the groundwater. So
That's true.
in fact, you get more nitrogen and whatever else you're getting from the groundwater. Does that again add value to keeping the pond open longer so this new input can be washed away? Or is that kind of opening the floodgates
Yeah.
to an endless torrent of nitrogen?
So yeah, I mean, just to explain it to some of the other people in the audience, so they know what we're talking about. It's all a matter of gravity, right? So the groundwater is flowing towards the ocean because there's a buildup of fresh water and it wants to go, let's say, downhill, right? Even though it's underground, but it wants to go to the ocean. If you open the pond, you're actually taking out the plug, right? The pond level goes down, and so the groundwater finds itself like too high, and that accelerates the flow of fresh water into the ponds. So the question, your question is, by opening by pulling the plug, we're actually making the groundwater flow faster and deliver the nutrients faster than it would do otherwise.
So if a cut closes too early, we see the pond actually fresher than we started before the
And more
cut,
polluted.
and sometimes more
That's it.
polluted.
So keeping the pond open for a certain amount of time is
Do we know-
actually positive.
Depends on the
Because-
pond.
Depends on the pond.
Okay.
But you really, those nutrients are gonna reach the pond, yes or yes,
Yeah.
right? So if you keep it open, even if you're putting a lot of nutrients into the system, like a little flush,
Mhm.
you can actually get rid of it if you keep it open, right?
Yeah.
Let it flush for a certain amount of time, and you will be able to remove that
Amount.
legacy that is still there in the
Yep.
in the groundwater.
And there's some comments from our colleagues at Georgia Pond in the Hamptons, Sarah Davison. We can thank them for an introduction to Dr. Gobler with cyanobacteria, mentioned that for them, and they've got a pond that's had more frequent and sooner harmful algal blooms, that IA technology is their main sort of way to combat nitrogen. And they're using Fuji Clean and Hydro Action as two of the most successful types with an average homeowner cost of $20,000 to $40,000. And in New York, they have town, county, and state grant incentive programs to help fund these. I think New York might be ahead of Massachusetts a bit in terms of capacity and funding. And so this might be another example where we can learn from the folks in New York, because they've been dealing with things sooner and faster. Things were more dense and more warm sooner. And I think Sherry might have had a question in the room.
Yeah, I have a lot of questions, but I have thanks. It was a great presentation. I wanted to let you know that the commission is working on a 208 off-island wastewater program. And we have updated all of the land use for the area. We've talked to the boards of health, and we're trying to put together for septic systems, what types are there and which have been upgraded. It's a painful process, but we've got a lot of information we would love to share with you.
Okay,
Um,
fantastic. That was
yeah,
my
I'd
follow-up to
love to
what
talk
you
to you
just
for like
said. I
another
hope that
hour
I
or
can
so,
have
yeah.
access. Oh,
Oh, yes, the commission is.
That's fantastic.
That
Thank you.
information is all available. How deep were you putting the groundwater wells around the perimeter? I found that was really fascinating. How deep were you putting in those
It's
PCometers?
basically a meter and a half. So we were
Wow.
taking advantage of the fact that being so close to the shoreline, the groundwater is actually like not very deep, right?
Mhm.
And as a matter of fact, the freshwater lens at that point actually like it narrows down and becomes like thinner because it's being pushed by the higher salinity water underneath. So we're taking advantage of the way freshwater flow to be able to capture very well the concentrations in the groundwater. So we didn't need to go very deep. And actually those rods that I was showing in one of the pictures, It's probably like 2.5 meters maximum, the longest that I have. And I only need to poke them down, as I said, 1 meter, 1.5 meters. In some areas, they may remember this, it was like super shallow, like the fresh water was like, sometimes you can even see it flow, right? Especially when there is like a cut in the inlets of some of these ponds, you can even see the fresh water coming out of the shore, right?
You can get the shearing effect.
Yeah.
Where if it's a really salty pond, there's a shimmer when fresh water
Yeah.
enters salt. One more question from the room. We are way over time. I know Javier's fun to spend the day with, but we've got time for one more question. Does anyone else have one in the room? Okay, anyone from the audience? One more question. If you unmute, go for it.
Can I ask a question? I think
Oh,
a really quick one.
Sam's got one apparently.
So when you were talking about Mashaka Cove, this is backing up quite a bit. And you were talking about putting more houses into the sewer system and then that being then a bigger input that was getting into the pond. But then it also sounded like getting houses off of their septics under the sewer system was a potential mitigation. Basically, what I'm wondering, thinking from a policy standpoint, is like more, I think I know, more houses is more houses, or is the sewer system, you know, if the sewer system in Edgartown is a source to Mashaka that is
Yeah.
so high, and we think, oh man, we're gonna solve a lot of this problem by getting all these houses in Edgartown in the sewer system, and they're all flowing to that. I mean, it looks like,
Yeah, I see.
it seems like
your
you're
point.
capturing it in one place, so as far as mitigation, you could really focus mitigation, but it sounds like the nitrogen isn't taken out of the system, it's just sort of corralling it.
Well, the wastewater treatment facility is doing a role of removing some of the nitrogen in that wastewater,
So
right?
it's downgrading the concentration before it releases it
Exactly.
into Mashaka.
Now, the more houses you connect to the facility, if they are not within the watershed.
And
of
there
the-
are many homes that are
Exactly.
currently connected to the wastewater treatment facility that are connected. And they're not in the Evertown watershed. So there's a huge number of homes that are outside the watershed and that is pumped in. The other thing is there are hundreds of homes with pipes in the ground in the watershed that could be connected to the wastewater treatment facility. Thankfully, the town of Edgartown is going through the comprehensive wastewater management plan, as are other towns. And right now they're looking at how to balance that flow. So we're not the only ones in the room concerned about it. This is in Catlow, the engineer on this from Ty and Bond is looking at how do you balance that. But it's, I think from a policy question, what needs to happen when, in what order, how fast?
Yeah.
That's
exactly correct, and I'm gonna advocate for my own project, but that's why, in a sense, it makes sense to update all this modeling and to be able to tell, where the nitrogen is coming from, whether, what happens if I move these septic systems into the wastewater treatment facility and what difference it makes, because it will help answer that type of question very well.
The other thing about the treatment center is that until really recently, I live in Oak Bluffs, but when my septic system got pumped, that waste went to the Edgartown.
So for your
facility.
individual
So for a long
septic.
time, Edgartown's been receiving the waste from everyone.
Yeah, we have a similar
Right.
problem on the Cape, in Falmouth where I live, right? The little pond in Falmouth, was for the most part like sewer, the entire area, almost the entire watershed sewer, which was like a very costly project. I believe it was, don't quote me on this one, but I think it was $20 million. They pumped all those, wastes to the West Falmouth wastewater treatment plant. So you actually move the pollution from Little Pond onto the West Falmouth Harbor. Now, the amount of nitrogen is not equivalent. We have this much here, and now we only have like 5–10% of that there. But it's a lot of houses. So when you add it out, it's a lot of nitrogen for West Falmouth Harbor that came from outside.
Well,
So that-
thank you very much. We should probably end the Zoom, but thank you.
Well, thank you.
In your office. Yeah.